Hello, welcome to my presentation today. This video will be streaming live at the Fall Search Seminar Day, and if you're interested in asking any questions or want to talk about the following research, feel free to stop by my booth at the Oral Presentations Q&A session. My name is Amanda Piapani, and I'll be talking about my research analyzing the nervous system of leeches. I worked with the Fegnar Lab at Caltech, and my mentors were Dr. Daniel Fegnar and Dr. Pegah Kasarayan-Fard. Within the scope of the Fegnar Lab, I was really interested in seeing how nervous system structure supports behavior. This covers neurite growth processes, synaptic clustering and generation of behaviors, as well as modeling neuronal activity patterns as a function of synaptic input patterns. If you're unfamiliar with neuroscience, the nervous system is composed of neurons that transmit information throughout the body. In order to do that, neurons work together in neural circuits and follow very specific pathways. Neurons generally have a cell body, seen here, an axon, and multiple dendrites. For leeches, there isn't a structural distinction between axons and dendrites, so we'll be referring to those as neurites. The structure of a neuron we're interested in are the synapses, which are the sites at which neurons relay information and are generally located at the end of an axon for a presynaptic neuron and at dendrites and the cell body for the postsynaptic neuron. The terminology we use in our lab to understand neurites is based off of graph theory. So first we start off our tree at the cell body, and then we make our way up from the neurites that grow from that cell body. A node is a branching point that connects adjacent segments, and a segment contains a node at its beginning and end. Parent segments and child segments are connected by a node, with the parent's last node being connected to the child's first node. As you can see here, we have a parent segment, and a child segment to that, and here a child segment to this parent segment. We have two types of segments. We also have intermediate segments, which contain at least one child, and terminal segments, which contain no children. So what does the field currently know about how neurite morphology affects output? So unlike a field like physics, we don't have a strict formula for where and why synapses form at the locations they form at. We currently know that neural circuits process information differently by the types of neurons and how their synapses are distributed. For example, studies over the past decade have shown that certain neuron models match biology better when synapses are randomly allocated than clustered and vice versa. In the rat, there's evidence that shows that hippocampal cells work better with randomly allocated synapses, and pyramidal neurons work better with clustered synapses. Today, we'll be focusing in on our work analyzing neurite growth processes. A growth process refers to how the segment develops from adolescence. We look for where and why synapses form at the locations they form at. We currently know that neural circuits process information differently by the types of neurons and how their synapses are distributed. For example, studies over the past decade have shown that certain neuron models match biology better when synapses are randomly allocated than clustered and vice versa. In the rat, there's evidence that shows that hippocampal cells work better with randomly allocated synapses, and pyramidal neurons work better with clustered synapses. Today, we'll be focusing in on our work analyzing neurite growth processes. A growth process refers to how the segment develops from adolescence. We understand the growth process through analyzing the distribution of terminal and intermediate segments. Once we get a better understanding of this, we can even develop a model of how the segments develop through simulating probabilities of features such as growth, branching, stopping, and retracting. This isn't an exhaustive list, but it's generally what most people model growth processes with. So here today, we're really interested in studying leeches, specifically the medicinal leech. We study leeches because as invertebrates, they first have a relatively accessible nervous system. Second, they have relatively large neurons that yield strong signals. And third, they express identifiable behaviors, which is really crucial since our trials are divided into different behaviors. We'll be focusing on the leech's DE3 motor neuron and 3 key behaviors: swimming, crawling, and an escape reflex we refer to as local bending. All of these behaviors share a common output in the DE3 motor neuron, and each of these behaviors' neural circuits include DE3. Our research primarily is focused around DE3R and its presynaptic partners. R just refers to the right. Here we can see the structure of DE3, shown in white here. Now in color, we can see each of its presynaptic partners and their structure. So in order to pursue our goals, we needed 2 key things about DE3. First, we needed to be able to record DE3R's activity during each of the 3 behaviors. And second, we needed to be able to record DE3 structure shown here. So how did we do this? First, we used voltage-sensitive dyes to record neuronal activity. And if you're familiar with action potentials, VSDs allow us to record the subthreshold neuronal activities instead of just the action potential. This activity is what we referred to earlier when we meant that leech neurons yield strong output signals. By using VSDs, we were able to show that synapses work in clusters for different behaviors, which we'll show on slide 18. So here we have 2 examples of a trace or output signals of a neuron using VSD. And this is measured through change of fluorescence over time. So on the left, we have the VSD trace of DE3R during swimming, and on the right, it's during crawling. And as you can see, these traces are visibly different. This tells us that the cell is operating differently during swimming versus other behaviors such as crawling. Second, we use serial block phase electron microscopy to anatomically image the ganglion where DE3R resides in. This technique was important because it also allowed us to get the exact location of the synapses. So here we can see the biological data that SBEM was able to record. And if you're interested in looking more, you can follow this link down here, leechem.caltech.edu, to look even further and be able to interact with this data. So here in green, you can see the tracing of the neurite of DE3. And if you compare it to the previous image, it's very difficult to see where these neurites should reside. This tracing took a lot of expertise and took up to 2 years just to trace DE3R. So today I'll just introduce the most important visualizations made during my research. So first we have the distribution of intermediate segments on the left and the terminal segments on the right. And we found that out of these 4 distributions, the log normal distributions fit both the intermediate and the terminal segment distribution the best by sum squared error. And so remember from the beginning, this was really essential to understand, to know the growth process of the leech. In addition, we also found terminal segments tended to be shorter than intermediate segments. And this could be because of a multitude of factors, such as the probability of retraction could be higher, or even the probability of branching could be higher in terminal segments. Second, we have the terminal segment length and edge distribution. And so we were really interested in seeing if synapses had a role in stabilizing the growth process. However, we did not find a correlation between synapses and if the growth process was stabilized or not. However, this graph was really important because it helped us find a measurement bias in our data, the first iteration of this graph. So if you look in the bottom left of these graphs, you can see an abrupt cutoff at 0.5 microns, where there seems to not be any segments with one edge and with length that tiny. And however, this was done on purpose. This is because we found we had an overrepresentation of terminal segments with one synapse of edge length 1. And this was because this was misrepresented in our database because what appeared to be terminal segments were simply synapses sitting on the wall of a parent neurite. So they were incorrectly labeled as terminal segments, and they should have just been located on their intermediate segment. This required a lot of data cleaning, and a really difficult decision to remove all this data here. We found change results as a result. We found change results in the previous graphs as a result of this data cleaning. Third, just a brief overview of some of the other work we did over the summer. We have a 3D model of DE3R and its synaptic clusters for the swimming experiment shown here. As we talked about in the VSD slide, there are different synaptic clusters for different behaviors because DE3 performs differently. So these clusters look completely different for the clustering experiment. Lastly, we also had an experiment where we were interested in seeing if we could predict the output behavior or the output activity of DE3R based on inputs from its presynaptic partners. And here it's the same units as the VSD trace. So thank you so much for listening in on my talk today. I've only been able to talk about growth processes in depth, but I look forward to talking about other subjects in the future. Thank you again to Caltech for sponsoring my undergrad research fellowship, and thank you again to my mentors, Dr. Daniel Wagner and Dr. Pega Kasarayan Fard, for the amazing opportunity to work with them.